Okay. Enzymes are globular proteins, and as such, they're very fragile. They're susceptible to pH changes, solvent changes, temperature changes. You're not going to have a solvent change, but certainly a temperature change or a pH change in our bodies. For example, our stomachs are pH 2. Our blood is around 704, I think. But all the enzymes are optimized for specific temperature and pH. Now, so what is an enzyme? Okay, an enzyme is a catalyst. What a catalyst speeds up the reaction. Okay. The speed at which a reaction happens has to do with this potential energy hump here in the middle. The higher the hump, slower the reaction. So if you can lower that hump, the reaction will proceed faster. Now, there's two considerations for a reaction. There is a kinetic consideration. That's how fast it goes. And also a thermodynamic consideration is whether it's favored or not. Now, I keep giving this example of the diamond and the charcoal. Okay, so thermodynamically, charcoal is favored, not the diamond. Kinetically, it's a horrendously slow reaction. So it's kinetically not favored, thermodynamically favored. And those are two factors in every single reaction. In other words, is the product stable or not? That's thermodynamics. Secondly, kinetics. Does the reaction speed come into play? So a catalyst is going to affect the kinetic part of that reaction. So that hump there, when we go from reactants to product, is the deal breaker. In fact, I gave a presentation on that reaction between charcoal and diamond. And when I did the energy curve, I had to make it tiny because the activation hump was so incredibly large. So speed, and that affects, like I was saying earlier, that affects speed of the reaction. Okay. Okay, from a thermodynamic perspective, this difference between the energy of the reactants, that's potential energy now, and the energy of the products has to be a net negative for it to be thermodynamically favored or spontaneous. And there's a lot of ways of measuring that. Usually in introductory classes, we could talk about potential energy, but really it's something called Gibbs free energy, G. And that G has got two things built into it: enthalpy, which is potential energy, and entropy. Entropy is the measurement of randomness. So both those factors come into play when we're talking about whether a reaction is spontaneous or not. Okay, so a catalyst has to be also regenerated. It can't be used up. If it's used up, it's part of the reaction. Catalyst has to be regenerated at the end. So if we're using a metal as a catalyst, frequently we use platinum or palladium or nickel metal as a catalyst. It can't be eaten up. It's got to be there the whole time. If we're using a chemical like an enzyme, it's got to not be destroyed during the reaction. It's very important. Okay, because enzymes are proteins, and as you know from last time, proteins are very complicatedly folded. And part of that folding, there's a part of the enzyme, or part of the protein called the active site. And that active site is where all the magic happens with the enzyme. So that active site is very, very, very specific. It can tell the difference between a D and an L form of an amino acid or D and L form of a sugar. It's very, very specific. Sometimes it only works on one substrate. For example, sucrase. Sucrase is an enzyme that catalyzes sucrose into glucose and fructose. It only works on sucrose, nothing else. So that's what they say absolute selectivity. That's what they're talking about. Relative specificity acts on related things. For example, amylase is an example of a relative specificity because it works on carbs in general. Not any specific carb, but just in general. And then they have stereochemical specificity, and that's the difference between a D and an L, for example. So these enzymes are really wonderful molecules. There's a lot of magic in an enzyme. One of the principal investigators when I was in graduate school, I was looking at, his lab was a computer lab. He didn't touch chemicals in there. All he did was he modeled enzyme activity sites using something called response surface methodology. And what that does is it actually mathematically models what that enzyme active site looks like. And then you change the pH, and then you look at the modeling again, how it changed and stuff. It was real interesting. He didn't have very much funding though, so I did not work for him. Okay. One of the nice things about enzymes also is they can be regulated. For example, when we need energy, we have enzymes that will become activated and start the glycolysis cycle. It'll start taking our glycogen molecules, breaking them into glucose, and that glucose enters into the glycolysis process and produces adenosine triphosphate, ATP. That's the regulation process. So if we mess up the regulation process, we really mess up our systems in our body. For example, fructose, we're going to talk about glycose for unit 5, but glycose has got a number of steps. The first 3 steps of glycolysis is where fructose comes in to the pathway. That's not regulated. Only the first 2 steps of glycolysis are regulated. And that's another reason why fructose is such a bad sugar, is because it goes into glycolysis unregulated. So even if you don't need ATP, you got tons of it. When fructose is there, it'll still be chunking out more because it's not regulated. Okay. Many enzymes require additional chemicals. They're either another protein or a non-protein. Things like vitamins, and they're called cofactors. Okay. Cofactors can be, as I was saying before, organic or non-organic. If they're organic one, they're called a coenzyme. Now, if an enzyme requires a cofactor and is without one, it's called an apoenzyme. In other words, it's missing its cofactor. Now, without the cofactor, the enzyme's not going to do its job. For example, if it needs a vitamin as a cofactor and you're deprived of vitamins, then that enzyme is not going to do its job, and you're going to be deprived of whatever that pathway where that enzyme is involved. As you can see, many of these are vitamins. Riboflavin, thiamine, vitamin B12, just a whole bunch of them. Lipoic acid. Those are all cofactors, and all those have an enzyme, an apoenzyme that it hooks up with. Now, in biochemistry, we don't call the reaction the same thing. Instead of reactants, we call them substrates in products. So we have a substrate, we have an activated complex called the enzyme substrate complex, and then we have products. And notice the enzyme is regenerated. Start with the enzyme, end with the enzyme. Okay, so we have substrate, product, all facilitated by an enzyme. Now, that is right here. That is where that activated complex does its thing, at the top of the activation energy curve. So this is on the left side of the graph, right side. And again, remember we have two factors. We have thermodynamically favored and kinetically favored. Now, there are many theories on how the active site works. One theory is called a lock and key. What that means is the substrate has a place on the enzyme called the active site, and it fits in there perfectly, like a lock and a key. The other theory, which is designed for more non-specific substrates, we have induced fit. In other words, the active site, when the substrate comes in, the active site will conform to the substrate, as opposed to the lock and key, where the substrate has to fit there exactly. Okay, there's some terms when we talk about enzyme kinetics. Activity. That's how fast an enzyme will turn over a reaction. This varies widely. Each enzyme is very different how fast it operates. It depends on many, many factors. But the process is called, and the numbers have to do with something called activity. To measure the activity is called something called a turnover number. And then when they do something called an assay, that's when they determine these turnover numbers. So some enzymes work really slowly, others are blindingly fast. Sucrase is a very fast enzyme, for example. As soon as you have sucrose in your mouth, it's converted to glucose and fructose. Literally that fast. It's a very fast enzyme. Look at these turnover numbers differences here. We have 36 million versus 900. Big difference in turnover numbers. So enzymes, you can't look at an enzyme and determine how successfully fast it's going to go or not. Okay, so we talk about the speed at which a reaction happens. These are the three factors that determine that. Okay, so concentration of the enzyme. More enzyme there, you get to turn over more. If you have more substrate, you're going to produce more product. And then you have these environmental factors that come into play that will affect the speed at which that reaction happens. That's solvent, which doesn't come into play in the body because it's just a water environment, doesn't change, but pH and temperature does change. That's why the body goes to great lengths to make sure both of those are well regulated. That's why a big deal when you run a fever is because your enzymes stop working. So the rate, how fast it's going, increase the enzyme concentration, the rate goes up. There's just more catalyst there to facilitate that reaction. However, it doesn't go on forever. So if we increase the substrate and we have a fixed amount of enzyme, then the rate is going to peak. It's going to flatten out. That's because there's not enough active sites to do the reaction. And again, solvent's a factor, but not in our body because our body solvent doesn't change. The only place it sort of changes in our stomach, but it's still an aqueous environment. What changes drastically in our stomach, though, is its pH. Temperature is very important. Globular proteins will change their shape on temperature and pH changes. If the globular molecule changes its shape, then the active site is going to change its shape also. So the substrate is not going to fit into the active site if the enzyme shape changes. Physical movement and temperature are closely related. Temperature is a metric used to measure the molecular movement. So if you're taking something like you're beating an egg white, what happens when you beat an egg white a lot? What happens? What does it turn into? It's fluffy, right? What do they call that? Meringue. Meringue is egg whites that you denature with physical activity. Okay. So the rate and temperature are very closely related. And there's always an optimum temperature and an optimum pH by which enzymes work their best. It's like pH. Now, if you've ever made a salad dressing, frequently there's raw garlic in there. And when you make a vinaigrette, you're changing the pH of that down to really low. So that's going to moderate the enzymes in that garlic that makes it harsh. That's why vinaigrettes are as harsh as just eating plain old garlic, even though the garlic is uncooked. When you cook garlic, you make it susceptible to temperature changes, increase the temperature. What that does, that modifies the enzyme that makes that garlic harsh tasting. So garlic now is becoming very mellow. That's why frequently you don't throw garlic in at the end of your cooking. You always throw it in towards the beginning so it can cook and that harshness goes away. because the enzyme is being denatured that makes the harsh flavor there. If you do change polarity, solvent polarity, the intermolecular forces will come into play on the amino acids functional groups. There are 20 amino acids, and they have 4 different classes. There's 2 kinds of neutral, acidic, and basic. So once you start changing the IMFs, then you start changing that interactivity between those side groups, and hence the shape of the protein changes. Okay, so series of enzymes here. That's a protease. It means it hydrolyzes protein. Proteases hydrolyze protein, and an example of one is pepsin, and that's located in your stomach. Notice the optimum pH. It's really low. These guys are typically what you see for enzymes in the rest of our bodies. In other words, sort of neutral, fluctuating around pH 7. Okay. So there's a number of different kinds of inhibitors that slow down the reaction rate of an enzyme. So in biochemical terms, it's called decreasing enzyme activity. But inhibitors slow things down. There are reversible inhibitors and irreversible inhibitors. Irreversible ones, once it happens, it's done. A lot of antibiotics and poisons are irreversible inhibitors. Poisons, for example, there's an enzyme called the cholinesterase enzyme. And what that does is that removes the acetylcholine as a neurotransmitter as it goes across the synapse. When you blast that enzyme, it cannot release the acetylcholine anymore, so the nerve cannot function anymore. It's frequently used in pesticides. And the class of pesticides is called organophosphates. Those are all cholinesterase inhibitors. Heavy metals, another one. That's why mercury is bad for you. That's why lead's bad for you, because it works with a cysteine amino acid and ties it up. So if that cysteine amino acid is part of an enzyme, the enzyme no longer functions. And the only way you get rid of that is to excrete it. Antibiotics screw up bacteria and viruses' enzymatic pathways by binding with certain things, like sulfur drugs, penicillin. It makes holes in the walls of cells of the bacteria. For example, cyanide. You all heard of cyanide poisonings being bad. What that does is it works on the electron transport part of the last part of carbohydrate metabolism. And everything just stops there. So you can't produce any ATP. Hence your body will just die because it has no energy. Mercury and lead ties up cysteine amino acid. Where is here? Penicillin works on cell walls. Makes the cell walls very weak and holes in them. This is the last enzyme in the electron transport series of the metabolism. And that enzyme is tied up with a cyanide. That means that enzyme no longer works. This is it here. Okay, so those are irreversible ones. The opposite ones are reversible. You can turn it off and on. That's what reversible means. Turn the enzyme on, turn it off. So there's a lot of different ways you can do that. One of the ways is to compete for the active site. So if this is the active site, and that's the substrate, that greenish inhibitor will fit in the active site first, preventing the substrate from coming in. So it kind of takes up the space. So the inhibitor has got to have a lot of the same characteristics of the substrate, otherwise it won't go in there. There's a lot of competitive inhibitors. A lot of drugs work this way. A lot of the sulfur drugs work this way. Some of them are considered antibiotics, for example, if they're penicillin. The other way inhibitor works is to bind on the surface of the enzyme, but not in the active site. Now, there's two ways to do this. There's an allosteric way, which I'm going to talk about in a little while here. So there's two ways to do this, where the inhibitor binds to the surface of the enzyme. And when that happens, the active site shape changes. So the substrate no longer fits in there. So when the inhibitor is bound to the outside of the enzyme, the active site changes shape and the substrate won't fit in there anymore. Oops, what happened there? Okay, there's something called a modulator that is sort of like what we just saw on the last slide. It can, when it binds to the enzyme, it changes the shape of the active site. And if it can either change it so it makes it more conducive for the substrate, or it changes it so the substrate can't fit in. So if it changes it so the active site is more conducive to the substrate, it's called an activator. If it changes the shape so the substrate can't fit in, it's called an inhibitor. And both kinds. And that's called allosteric regulation. So whenever you have allosteric regulation, the enzyme has two active sites. Two active sites. One is for the modulator and one is for the substrate. The modulator site is designed also very specific, so that only accepts the modulator's size and form. Once it fits in there, then something's going to happen to the active site. If it's going to inhibit it, it's going to change it from something where the substrate can fit in where it can't fit in now. So that's an allosteric inhibition. And the opposite is true. If the active site is not conducive to the substrate coming in, but once the allosteric activator is in the other active site, it changes the shape of the active site so the substrate can then fit in there. So that's considered activation. So there's allosteric inhibition, allosteric activation. Many enzymes run this way. They have two active sites. One's for the modulator, one's for the substrate. There's yet another one. It's called enzyme induction. And that's where the system actually generates an enzyme based on the requirements. So for example, here, if we needed to take this lactose disaccharide and hydrolyze it, it's going to require that enzyme. That enzyme is not naturally there. So what happens, our body makes it on site, and hence it works. So that's called enzyme induction. Now, all these examples, you don't have to memorize. They're just examples. You have to memorize the processes, though. All right, so as far as enzyme inhibitors go, there's irreversible ones where it's dead in the water, kills the reaction. Then there's reversible ones. There's a couple different kinds. There's competitive ones, non-competitive ones. Competitive ones will sit in the active site to prevent the substrate from coming in, and the other one works by allosteric regulation. Okay, so there's going to be two presentations on enzymes. Okay, there's this one. And there's also one on naming enzymes, and it's based on what the enzyme is doing. So that's what we're going to talk about next. Looks like I didn't load it. Where is it now? Well, this is embarrassing. Good thing I know you guys know. Where'd you go, dadgummit? Okay, what we're going to do is okay, so let's look at the practice tests. This is what I was going to do Thursday, but let's just go over this today. Okay, this is an in-person practice test. It was one I gave last semester, and it included one question I did. We're not going. It's included, but we haven't covered it today. That's not a name an enzyme, since I couldn't find the slide to cover it. Okay. All right. So going back to Tuesday, and we talked about when proteins fold, there are crosslinks that determine the shape of the resultant protein. And there are 4 different strengths of the cross-links. Okay, so strongest, weakest. The weakest one is only intermolecular forces, and it's LDFs only. So it's very weak. So it's very susceptible to temperature, pH changes. The covalent one, the disulfide, is very strong. It's not dependent on temperature and pH. It will stay linked up. So where I was going with this question was basically is which crosslink would be most susceptible to solvent change? Okay, solvents are involved in intermolecular force interactions. So that would mean this one would be least sensitive. And why? Because it's got the strongest or the weakest intermolecular force attraction. Okay, you're not going to see a question like this. It's going to be, instead of all in one like this, I would break it up. But basically, here's an amide. How do you make that? Here's a secondary alcohol. How do you make those? Here is an ester. How do you make that? So we're going to go through these examples in a minute here. But whenever you see an amide, how do you make it? There's 2 ways, with the acid chloride and with the anhydride. Same with the ester. And to make a secondary alcohol, you can either reduce a ketone or oxidize it. Not oxidize. You can reduce a ketone or put water across a double bond. You'll get a secondary alcohol. Okay, so name this guy. Remember, there are 2 ways to name an amine. So we have an ethyl group here, and we have a T-butyl group here. So one way to name it would be T-butyl ethylamine. That would be common. Yeah, UPAC, we have to find the longest backbone, which is going to be right here. So that's going to be a propyl. We're going to have 1 methyl branch and the amino branch. So it's going to be 2 2-NN dimethyl amine or amino, and two methyl propane. That would be the IUPAC. Now, we didn't stress T-butyl too much in this class. I did last semester, though. But anyway. Okay, does that make sense on the NN dimethyl. That refers to these guys here. Okay, so we need to make an amide now. Remember the two ways again. So what does this guy look like? All right, well, it's got buta means backbone of 4. So we have okay, so we have dimethyl, NN dimethyl. That's the amine part. Buta, and that refers to the acid part. So this is the acid. This is the amine. So to make that, we start with the acid chloride. Plus the amine. That's one way. The other way is with the anhydride. Oops. Now, starting with the bare acid does not work with the amides. The reaction is extremely slow to the point where it doesn't happen. Esters, you can get away with it, not amides. Okay, does that work? All right, so we have the amine, the acid. Okay, so the acid's a 5 backbone acid. And with a methyl branch. And we have one methyl group sticking out of the nitrogen. So when you say dipeptide, you need to draw 2 structures because half the time it's going to be, well, yeah, no, excuse me, 1 structure. Because it says leucine goes first, then glycine. But if you just took leucine and glycine in a test tube, you'd get 2 versions. You'd get one where the leucine's first, or the glycine's first. Or the, yeah. Okay, so okay, 8 carbon backbone. The amino group is going to be carbon 3. The isopropyl is going to be in carbon number 2. Common biological molecule, that's an amide. That's oh, what else? Insulin. By the way, why do you, Why did type 1 diabetics can't take a pill? They have to give a shot. Anyone know? Shots are a pain. I mean, they're painless going in, but they're just, you have to get the syringe out and all that junk. Why do you think they just can't take a pill? Like type 2 diabetics take pills. Why do you think? I just mentioned that insulin is an amide. It's polypeptide. What happens in your stomach with polypeptides? Hmm? They get digested. Yeah. So if you took a pill of insulin, it, huh? No, because the pepsin digests it and breaks it apart. So it's no longer insulin. It's a bunch of amino acids. It's treated just like a piece of meat. So it's really imperative to bypass the stomach. That's why you have to inject it. So whenever, if you have any friends that are type 1 diabetics on insulin injections, that's why they have to do an injection. Sometimes it's a pump or physical injection. So it's too bad, but it's just the way our stomach works. Anyway, there's a lot of answers to that. Any of the DNAs or RNAs are all amides. Insulin, there's a whole bunch of them. Okay, name the enzyme responsible. Okay, we haven't gone, that's what I was trying to find. So I'm going to go over this on Thursday. Basically, you look at what's going on in the reaction. In this reaction, okay, so this would be a glucose kinase. So kinases put phosphate groups on molecules. And this is the first step of glycolysis act. So you take glucose and it's phosphorylated so that it becomes a glucose 6 phosphate. And so what you do is you name the substrate and then the action. Okay. Now there's a presentation on this for Thursday that I couldn't find today. Okay, this one here, our substrate is going to be 6. Okay, 6-phosphoglucose. That's a substrate. And what we're doing, we're converting it to fructose. So that is an isomerase. So that enzyme is named 6-phosphoglucose isomerase. Okay, what's a coenzyme? Coenzyme is required with a apo enzyme. So you have an apo enzyme that requires a cofactor. And if it's an organic cofactor, it's called a coenzyme. So once those come together, it forms the enzyme. Okay, how can we control an enzyme? And you just go through what we just talked about. There's allosteric. Okay, there's allosteric regulation. There's competitive inhibition. And when I say control, that is inhibited or activated. Okay, both these can be done allosterically. These are both inhibited. And what factors contribute to enzyme function? pH and temperature. You could write down solvent, but it doesn't come into play if it's a mammalian biochemistry system. And we talk about allosteric regulation. We are talking about okay, it's a method to control the rate of enzyme activity through activating it or inhibiting it via a second site. On the enzyme. Okay, and that was the test. Okay, so that's what I wanted to cover today. So I'll go ahead and end this.